Good afternoon, guys. If somebody could type in that Padlet and let me know if you guys can hear me, I'd greatly appreciate that. Okay, so I see I've got a few from 391, one from 393. If you guys will let me know if the video's showing up, that'll help me out because I'm live streaming to all 4 sections simultaneously. And while you guys are letting me know about that, I'm going to run and grab my water and turn the other computer down. All right, so the Padlet is going to be our way of communicating so that I know how this is working. So if someone from one of each sections could just let me know if this is playing through so that I know that you can see me and hear me. Have you pushed play on the video? Hold on. Okay, let me check the... So you guys, is anybody hearing me or seeing the video now? Okay, I'm seeing a comment that I can see the video now. Can you tell me what section you're in so that I know that as you guys comment on it, because I'm trying to stream all 4 sections simultaneously. So if I can know that there's someone in each section that's seeing it, that'll make me feel a lot more comfortable. All right, Blake, I know you're in 392, so I know 392 seeing it. Do I have anybody in 390 or 391 that's seeing it? Okay, as you comment, let me know which. Okay, I'm seeing a 391. I've got someone in 392 that's telling me they're seeing it. All right, so connection could either be your internet speed or it could be my internet speed, so that is beyond my control. And I'm actually not at home. I've got a setup that's better than at home, but my camera's not come in and the phone's still not working to live stream. And I apologize, I'm not looking at the camera. I'm busy reading all your comments that are coming in super crazy fast. Pastor and elders gave me permission to come in so that we could do this review and utilize the church media. So if I disappear, I've gone to the other computer because I'm trying to be a couple people in one place by myself. And part of the problem with some of the internet and Canvas, all these tools, is that all of the teachers across the nation are trying to go online and do various techniques to help their students. So the beauty of this is even though you might have lag time now, it should record it so that it plays continuous a little bit later if you have to go back to it. Okay, got quite a few of you. Okay. So the Padlet is so I can try to keep up and see if you guys have any questions. And try this format out. This is completely new to me to do this because I'm used to being in the classroom with you guys. And the conferences is an option as long as I'm trying to work with one class at a time. And that's part of the problem that I'm going to have continuing the face-to-face. I'm not going to be able to keep the 391 and 392 going simultaneously. Conference is linked to individual sections in Canvas. And so over your extended break / my work period, I was working to find a way that I can actually present to all 4 of my classes simultaneously and do some review sessions since we're not going to be face-to-face. And you'll be able to see my face, but I sure hate that I can't see your faces and know if you're getting it, if those lights are coming on. So you guys are going to have to communicate with me via messaging through the course questions or material discussion forums, or when we're doing these sessions like this where I try to bring all 4 sections together, post in these Padlets, and I'll do my best. In the future, I won't have you guys all put your names in on a review. It's just today is to help me kind of see what kind of turnout I'm getting to see if this is going to be a worthwhile tool to actually utilize moving forward. And then what I'll do is I'll kind of pick a time and do some review sessions, at least one review session each week. It'll probably be on a Monday afternoon because that's the most open time for me. And so what I would encourage you guys to do is, if you do have questions and you cannot attend the actual session while it's running live, pass those questions off to a classmate that may be attending the class. And another thing I'm going to do is I'm going to have you guys, I'm going to put another discussion forum in where I can ask you guys to post the questions early so I can kind of come in prepared, since we are going to have some lag time between me speaking and then you guys receiving it. In fact, it's enough lag time between the time I talk and the time you receive it that I can walk to the other end of the church and come back. So I know that's going to take a little bit. When I ask a question, it's going to take a little bit of time for it to reach you guys, and then you guys respond back to me. So the first thing I want to do, and you'll have to pardon me because I'm having to deal with these microphones too, is I wanted to go over fermentation. With all the changes and all the stuff that's had to happen in the last week to week and a half, because I was waiting for the final word on what was going to happen. I didn't want to create a schedule and then be told, oh, we're going to do it this way instead, because final word was going to come down on the 24th as to what we thought was going to happen. And so we got word then that we were going to be closed the rest of the semester, and I had an a better idea of what I was working with. So I've spent last week trying to come up with ideas of how would we meet the face-to-face component, how would I be able to interact with you guys to do some review sessions so that you could see the drawings and stuff, because it's going to be very difficult to do that kind of thing with conference. I'll be able to talk to you, I'll be able to share my screen, but you won't get the drawings and things like that you're used to in the lecture hall. So that's where we'll do those kind of things in review sessions. And one of the things I have been working on, have a transcript for even, was a fermentation video, and just the time has escaped me to be able to sit down and get that accomplished. Now, it's a little bit flimsy. It's that washboard like we work with in the classroom. All right, so with fermentation, the key things that you need to understand is what I'm going to focus on here. I don't have beautiful PowerPoint or all kinds of lovely pictures to help you out. So we're going to do some real simplistic drawing and go over the basic concepts of fermentation. What I've drawn here, hopefully you guys recognize, I mean, your test is coming up tomorrow. Hope you're recognizing that I've drawn out just a few pieces of glycolysis. So here at the top we have glucose, then we have our energy investment phase, our lysis phase where we make glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. We have the isomerization where we would end up with 2 of those G3P molecules, and then this is the area right here that I want to focus in on. So there's a number of steps that I'm not showing the details because I want to focus actually on this right here. We talked about in the lecture hall, we talked about what we did with this NADH when we were running something like aerobic cell respiration or anaerobic cell respiration. Well, not all cells have electron transport chains. And if they don't have an electron transport chain, they're not going to be able to take this NADH and remove that hydrogen and oxidize it back in to NAD. And so this would be a problem in a cell like that. And this is where sometimes when your statement's said in class, we don't always do a good job of clarifying what we've said. And we're real bad about it when we're teaching A&P. One of the statements that gets said that makes it so confusing for what I teach, and it's usually said, you hear it in A&P or basic biology first, is that fermentation happens in the absence of oxygen. That's a true statement in some situations. But in other situations, it doesn't hold true. So let me give you an example so that we can talk about the importance of why this has to happen. There are some cells that we call strictly fermentative. That means they only do fermentation and absolutely nothing else. An example of a strictly fermentative cell is going to be a mature red blood cell. Red blood cells lack a nucleus, but they also lack another important organelle, an organelle known as the mitochondria. And at this point, you guys are already familiar with the fact that mitochondria are needed to be able to run cell respiration. Now, the reason why we don't utilize the mitochondria in the erythrocyte is because that erythrocyte would actually utilize the oxygen en route to the cells that it's supposed to deliver it to. That would be bad. So you imagine those red blood cells showing up at the toes and like, sorry, I used your oxygen, I'll be back. We don't want to give this guy a mitochondria. And so because we've taken the mitochondria out, there is no option for aerobic cell respiration. And so there's no citric acid cycle, no preparatory step, no cellular respiration. And so this type of cell, while it's in your human body, is what we call a strictly sorry for my handwriting here, fermentative cell. There are other cells in your body. So you guys probably, if I start to draw it out, you'll recognize it real quick. Multinucleate cell has striations, nice and elongate, called a skeletal muscle cell. They have an abundance of mitochondria. And so they will run aerobic cell respiration and continue to do so until their oxygen supplies are low. And in that type of cell, in the absence of oxygen, they would switch to fermentation. So in this cell, that would be a true statement. In this cell, that statement would be false. So, and think about it. The reason why I'm bringing up the erythrocyte's job is to carry oxygen. Its job is to transport that oxygen being bound to hemoglobin. And there's approximately 280 million hemoglobin molecules in a single erythrocyte. And each one of those carries 4 oxygen. So there's plenty of oxygen available in the erythrocyte. The reason why it's strictly fermentative is because it lacks the enzymes and lacks the structure needed to carry out cellular respiration. So what happens is this begins to accumulate, this NADH. Look at the problem with having NADH accumulate. We talked about these as being electron trucks. We equated them to like rock trucks. You don't get to just make a new rock truck if they're all full and you need your driveway fixed. You've got to wait till one becomes empty and available to carry a load for you. So here, if all of the NAD gets reduced to NADH, we're not going to keep running glycolysis, and therefore we wouldn't make any ATP at all. So the process of fermentation is going to be the events that happen after we form pyruvate. So what we see happening after pyruvate in a cell like a bacterial cell or our human cells, the skeletal muscle cell, this NADH would oxidize pyruvate to form lactic acid. And in doing so, the NAD would become, it's reducing the pyruvate, it's reducing the pyruvate, and the NADH is being oxidized to NAD so that it can return to the glycolytic reactions. Now, let's think for a moment. We have 2 products that were just produced from this reaction that's known as fermentation. So this is fermentation. We produced a molecule of NAD, and we produced some lactic acid. Now, for a moment, don't do this. We're supposed to be social distancing. But imagine for a moment you could actually leave the house and go to the gym. And you crawl up on one of those machines, and you set a timer because you're going to be a little bit ambitious. And you decide that you are going to start to do some leg extensions. After a while, it starts to burn. That burn that you feel is because your cells are having to switch over into fermentation, and they're producing lactic acid. Lactic acid is not the product that we're seeking. Lactic acid is altering the pH and can be detrimental to the cells. We were running that process because we were looking for this NAD to be produced so that we could continue to run glycolysis. That is no different for any cell, so it doesn't matter whether it's the bacteria that's run the fermentation or whether it's our erythrocyte or any other cell. If it's running fermentation, it is because it is trying to regenerate NAD. Now, when they do this, not only can they go straight to an acid, we call them organic acids because they're organic molecules, but they can also kick off carbon dioxide. So we'll see some bacteria producing a gas product and an acid, and that's why in the carbohydrate fermentation test that we learned in the first unit in lab, we could put a pH indicator in the test tube with that single sugar, and then as that sugar is being converted into that acid, it's changing the pH. And I know this process, this step is happening, that we're getting to this acid product. And if it's producing a gas, then that gas is going to come off and gas is going to rise. And so the little Durham tube that was in the test tube was going to collect and trap any gas that was produced. So we were able to tell if a bacteria strictly produces the acids or if it is an acid and a gas producer. Now, yeast do things differently. So I'm going to ruin some of your weekend habits. In yeast, they will kick off a carbon dioxide. They will oxidize the NADH back to NAD, and they will make a product called ethanol. Now, ethanol and lactic acid are not the only two products that are made by microorganisms. There are other organic acids like propionic acid and the acetone that we use in the nail salons and in the lab. Those are also products of fermentation of microbial specimens. But the key thing that I need you guys to recognize is what is the purpose of fermentation. And the purpose is to regenerate NAD so that glycolysis can keep running. The products are waste products for the organisms. We seem to enjoy waste. And so there's a little bit of fermentation for you. Now I'm going to give this a little bit of time on the lag to see if it's reaching you guys. And I'm going to reach out to see how the review is reaching you guys. See if you're able to hear me clearly and see if the information is coming across. So somebody will give me some feedback on this, and then what I'm going to do is kind of watch. Because now what I'm going to do is ask you to give me some questions. And if the questions are on the review sheet, make sure you give me the number so that I can find it on the review sheet. Okay, so Rachel, yours is, you're seeing the video after you refreshed it, but I'm seeing a lot of people are having lag time. All right, so I'm hearing it's better if you're watching it on YouTube. So is it lagging in YouTube? All right, so here's what I'm going to do. Since it's lagging so much, we're going to put a discussion forum in. Actually, we're going to use the discussion right underneath the announcements, and you're going to type in questions, and then I'm going to come back in and record it, and then you should be able to come back and play it after it's been recorded to save it from doing the freezing. And we'll try looking at it in YouTube. Oh, there we go. We're getting a question. I'm going to stay on here for a minute. Question 118 on the review. All right, guys, so I'm hearing it's not lagging on YouTube. So if you want to switch and go out from Canvas, click on it at the bottom where it says YouTube. Go out and go to YouTube, and maybe it'll play more clearly. And we'll work on this to try and work kinks out of it. All right, so 118 on the review. Go to it. And then if y'all continue, if you've got questions on the review, put them in there, because it takes a little bit of time for them to come across. All right, so number 118 on the review. Why was the result of Griffith's fourth experimental scenario surprising, and what did this scenario demonstrate? All right, so what happened with Griffith's experiment is that he was working with two different strains of Streptococcus pneumoniae. He was working with a strain that had the ability to produce capsules and a strain that did not produce capsules. And so if you'll recall from Unit 1, we talked about the capsule acting like camouflage for the bacterial cell. And so it's basically what it was doing was hiding the bacterial cells so that it had the ability to get past the phagocytes in the immune system. And so in his first experiment, in the first sets of experiments, what he did was he introduced the capsule-forming bacterium to the mouse, and of course we would expect it to die. The mouse did. And then in another experiment, Griffith took the what was called the R strain, and which cannot produce capsules, and he put it in a mouse, and because they don't have that ability to hide, we're not expecting it to kill the mouse. Now, the next step he took is he heat killed. So he destroyed the S strain that had the ability to make capsules, and he made a mixture of the S strain with the living R strain. And so what we were expecting was, because the S strain was killed, we were expecting that putting this live R in with this dead S, we would end up with a living mouse. Well, lo and behold, the mouse died. And so scientists would chalk that up to maybe it was contaminated. And so then he ran the experiment in a test tube, same experiment, more controlled. Maybe the mouse died from something else other than the Streptococcus pneumoniae. And so he replicated the results in the test tube, and when he did, it was very surprising, because what he found was that that R strain, that living R strain that did not have the ability to produce capsules, was gaining that genetic information from its surrounding environment. And so when cells can do that, we call them competent. That means that they're taking DNA from the environment and taking it into their cell, through their cell membranes, and then expressing that genetic information so that they now have a new genotype, but also a new phenotype. So they're going to look physically different. So what was an R strain bacteria is becoming S strain. And the best layman's terms way that I can put that to you is, of course, I have a fascination with green eyes. My husband has beautiful green eyes, but I've got brown eyes. And so imagine if, and I hate to say this in this day and time, but imagine something happened to Britt, and all of a sudden those genes are no longer needed. This is usually the example I use. It's a terrible time to be using it. But imagine those genes are no longer necessary. And if I were a competent bacterial cell, I would be able to take those genes, take them into my cell, and then express them and have green eyes. Now, what's interesting is we've analyzed the genetic information of bacteria that can do that, and there are some bacteria that are competent that actually are carrying around pieces of human DNA within their genetic information. Not that they're expressing it or using it, but it's evidence that they have been able to accomplish this process called transformation. So hopefully that answers or clarifies question number 118 for you guys. So if you'll go in there on the Padlet and say, yes, 118 is clarified, or no, I need some more, that'll help me know what I need to discuss. All right, so I'm also seeing on here question 100. What are introns and how do they impact protein synthesis? All right, so usually I only talk about introns in eukaryotic cells, and if you'll pardon me for a moment, I am out of my realm. I have no eraser, and I don't want to erase that whole thing with my hand, so I'm going to grab a napkin or a paper towel and erase that so I can kind of show you what introns are in kind of a layman's format. Now, the definition of an intron is that it's an intervening sequence of genetic information that is it's going to be located in between what are called exons. Exons are what's going to be expressed. So let's do something that's very layman's terms to drive this point across. All right, so looks like a bunch of random alphabetic letters up there on that board, and I sure hope you guys can see those. From my end, it looks like you can see them. Let me get out of the way of the last of it there. All right, I promise there's a message in there. But unlike you, you don't get to sit back in your chair at home. The ribosome's got to bind and read. And if the ribosome was to bind and read this, it'd be like, what? But there is a message hidden in there, and guys, and they're in Thursday group, this is not you, yours is still Thursday. But here's the message. Your exam is tomorrow. All of those sections that I highlighted in green are what we call exons. They are segments of DNA to be expressed. So the message that I want to convey, the message that I want you to hear or to receive. And being that exons are part of the DNA, the genetic information, that information that's going to go out, that's going to be expressed, is going to be coding for amino acids that make a protein. Because genes are segments of DNA that code for a protein product. And so the exon is the expressible portion of this. The information that's located in between are what we call introns. They are intervening sequences that disrupt the flow of the genetic information. Now, we focus on that in the eukaryotic cells in our unit and for our exam, and we pay attention to what has to happen to them. And to help you understand, those introns are acting like bad sections of an electrical cord. And if you know a good old country boy, they're going to fix that cord by cutting out that bad section, and they're going to splice it together and put a little electrical tape around it. Well, your cells have a special enzyme. It's such a cute name. It's called a spliceosome. It binds to the mRNA, and what it does is it literally cuts out and splices together. So it's going to cut out the introns. And then it splices together the exons so that you have a fluid message. So that's what your introns are. They've got to be cut out, and then that mRNA molecule can be in a eukaryotic cell. It can exit the nucleus and then move out into the cytoplasm to be bound up by a ribosome and then expressed. So that is question 100. Let's see what other questions you guys have got on here for me. All right, 141 and 142. What enzymes are used for transcription? All right, so transcription is pretty simple. With replication, you had to learn about helicase, binding and unwinding and unzipping, and then primase, binding and laying down a primer, and then we talked about DNA polymerase 3 and DNA polymerase 1 and the ligase, and we talked about DNA gyrase and DNA the topoisomerase. So replication is very highly involved when it comes to enzymes. Transcription, much more simple. There's one enzyme, RNA polymerase. Let me write that across the top of the board. So RNA polymerase, when you hear the word polymerase, ACE, we're thinking enzyme. Polymers are large molecules, and so a polymerase enzyme will build large molecules from smaller subunits. Here it's telling you that the molecule it's building is RNA. So your answer to question number 141 is RNA polymerase. And then number 142, what are sigma factors? Those are going to be proteins that are going to be interacting, helping RNA polymerase bind to the promoter region on the gene to allow a bacterial cell to have it bound and positioned so that it can actually carry out the transcription process. So the cell type is bacterial, and it's proteins that assist the RNA polymerase. So hopefully that answers those two questions for you. All right, so we've done question 141 and 142. We looked at 100, and I've got someone now that wants to take a look at question 7. What are ribozymes and how are they different from typical enzymes? All right, so the name itself should give you some hint. When you hear ribo, you think ribosome. So a portion of the ribosome comprised of the rRNA is actually acting in an enzymatic fashion. It's acting as a catalyst. And so where most proteins, or most of your enzymes are protein in nature, ribozymes are going to be rRNA in nature and exist within the ribosome. And then I see a request for number 107. Keep the requests coming because that's the last one I'm seeing on here. All right, so because we're cutting things back, I'm not going to be asking you guys any questions over 106, 107, any of the post-translational, post-transcriptional regulation. We're going to get to the meat of what's really needed for you guys as allied health professionals. So you guys can mark number 106 and 107 off of your review sheet. But I'll go ahead and talk about both of those. Pre-transcriptional regulation, it's going to keep you from wasting all that energy of making the RNA and then making the proteins. Post-translational, you're regulating the protein by either, and there's a number of ways of doing this. You can phosphorylate it or you can remove a group from it. So in A&P, you guys learned about things like pepsinogen becoming pepsin. So pepsinogen with a proportion cleaved off of it is pepsin and then it's activated. That is an example of post-translational regulation. Pre-transcriptional, we're interacting with the gene to turn the gene on or off, and so that saves us a lot of energy and saves us a lot of building blocks that we're not wasting otherwise. So that's what those answers would be to those two, but we're going to eliminate those from the test. There are no questions over those on the test. All right, so I'm going to wait for a moment. See if we've got any more questions that y'all want to go over. The last one I've seen posted on there is the 107, so anybody else got any more questions? All right, so question number 8. List at least 4 different factors that affect enzyme activity and describe how they do so. All right, so in your textbook, there's a whole section that's all it talks about is factors that affect enzyme activity. One of the most important factors that you guys need to know, because we're going to continue to talk about it as we move into the next units. In fact, in this very next unit, Unit 3, we're going to talk about the importance of temperature and how it affects enzymes, because we're going to use temperature, and that's what I was doing every week when I was running the autoclave. I was using temperature to destroy proteins, and by destroying the proteins, I was disrupting metabolism. And so temperature is one factor that affects enzyme structure, thereby affecting enzyme activity. Another factor that affects enzyme structure and thereby enzyme activity is pH. With changes in pH, because some of the R groups that are associated with the each amino acid, they're charged. When you change the pH of the environment, you're affecting how those R groups interact. And so a change in either direction to acidic or basic, you could have those R groups moving away from one another. And when they do that, then you disrupt the three-dimensional structure the protein, and therefore you disrupt its function as well. So temperature and pH, and then of course substrate concentration. You're going to have a set number of enzymes, and then as you add substrate, if only one, so let's say we've got 5 enzymes, just for simplicity. We've got 5 enzymes, and we've got 1 substrate molecule coming in. So our enzyme rate, we'll say, is 1. But as we add extra substrate, we can increase the reaction rate because we have more enzymes functioning, but we're going to max out to a certain point. So enzyme concentration, substrate concentration effects, and then another important factor that you should be prepared for on the test is discussing inhibition, competitive and non-competitive inhibition, and we went over those in pretty good detail when we met in class. So those would be the factors that affect enzyme function and structure. And yes, guys, I've just got a question on here. Where are you reading questions from? I'm reading them from the Padlet. That way I can see all of them simultaneously, because I've got one screen out here in the hall in the church. And so part of it is in there. And so I'm running the Padlet to see the questions that you got. All right, so number 22, just saw 22 pop up. A bacterial cell surviving on a diet of lipids can still make carbohydrate polymers. How is this possible? All right, so let's flip this whole board. All right, so this is where I get to tell on my husband. He watches this video later, he'll enjoy this. All right. You guys have already heard me in some of the sections talk about excess sugars get converted to lipids. Lipids can also get converted to excess sugars. So my husband had seen this cute little, the little pictures on the internet, thanks to our daughter, that said, hold her close and tell her she's beautiful. If she growls, move to a safe distance and throw chocolate. Apparently, in times like this and in the summer, I get a little bit stressed out. And so here's what happens. I'm going to show you amphibolic pathways that allow cells to switch between 2 different types of molecules. So let's say we've got that excess glucose coming in. So with the glucose, we can take 2 molecules that are called DHAP and acetyl-CoA, and if I take that phosphate off of DHAP, I have a molecule called glycerol. Take this acetyl-CoA, reverse beta oxidation, and you're going to get fatty acids. Do that enough to accumulate 3 fatty acids, and if we combine those 2 together, we get what is called a triglyceride. So what I did is, the first thing I did was show you how this carbohydrate catabolism is tied to lipids, where if we've got too much carbohydrate coming in, how we can switch that carbohydrate to lipid. Now let's think about how could a cell that doesn't have a source of glucose, We can't make G3P. We're not making this DHEP at the moment from sugars. But we've got an excess diet of lipids. How could we still get some energy in metabolism? Reverse everything I just showed you. Use lipases to break the fatty acids off of the glycerol. Take the glycerol, add a phosphate to it. Make DHAP and use it in glycolysis. Take the fatty acids, carry out beta oxidation, and as you carry out beta oxidation, what you're going to produce is acetyl-CoA for the citric acid cycle. And so that is going to be your answer to that question of a cell surviving on a lipid diet. What you would do, you would just reverse that process that I showed you. Convert. Break the lipid into glycerol and fatty acids, convert glycerol to DHAP, convert the fatty acids into acetyl-CoA. All right, so 22, a couple of you asked about that one. We've got question number 7 done. Okay. And those of you that are tuning in that were struggling, the video will have recorded everything from the beginning. If it doesn't, if I see that it didn't, what I went over was fermentation. I'll come back and I'll do that again in a separate video. Wow. Okay, any more questions coming from the review sheet? All right, so termination of transcription and the difference between self-termination and row-dependent. All right, so row-dependent is a little protein that's going to bind to the mRNA. I really wish I had a PowerPoint right now. But you've got this molecule. I've got very limited line here. So imagine the DNA helix opened up. And this RNA molecule attached to the DNA dangling off. With a row molecule, it will bind to the RNA. What it's going to do is it moves up the length of the mRNA and it wedges. It acts to push and separate the bonds that are located right here between the RNA and the DNA so that it separates. So it's a protein that functions in that fashion. Self-termination is built into the DNA sequence. And what I mean by that is when you look at your adenine and your thymine connection versus your guanine cytosine, they have differing numbers of hydrogen bonds. So when you look at AT connections, there's 2 hydrogen bonds, and GC you've got 3. And so what happens, it takes longer to make more bonds. And so if you have a region where you've got a rich GC rich region followed by where we're making the bonds and then we speed up and then we slow down, speed up, what we're going to do is we're going to create a loop inside of the enzyme that's bound. And that loop is going to cause it to wedge and cause the enzyme to actually come off of the molecule. So that's the key difference between the two. One, we're using a protein, and the other one, we're using a sequence built into the DNA molecule to help us separate them. All right, guys, so I had this planned for about an hour. I've got another conference coming up at 3 o'clock. I'm going to be a few minutes late for. So what I'm going to do, I'm going to take the questions that you guys have got. I want you to go into the announcement. I know many of you have gone out to YouTube now to watch this, I want you to go back to the announcements page and let me set them very quickly. Or actually, keep posting them in the Padlet. Keep posting your questions in the Padlet, and then I'm going to come back in when I'm done with my other conference, and I'm going to answer some more of those questions when I'm done with the conference that I have at 3 o'clock. So don't stop posting. Post the questions. Keep them coming, and then that allows that time for me to see them and then come in, and then as I start the next session, then you guys can post a few more. So we'll start back up again. Let's say, for time's sake, to make sure I've got plenty of time, we'll start at 4:30, and I'll do about a 30-minute session from 4:30 to 5 o'clock. So I'll see you guys again at 4:30.